Today, I'll be explaining the life cycle of SARS-CoV-2, the virus that causes COVID-19. I will talk about how the virus multiplies in the lungs, how it kills lung cells, how drugs could be developed, and explain how some of the drugs being studied right now can kill the virus. SARS-CoV-2, like other viruses, is not alive, but is just a particle that travels until it finds a host, the right cell type that can help it multiply. Like other coronaviruses, it also has proteins on its surface, which are these spikes around it that make it look like a crown or a corona. These spikes attach to particles on the host surface, fitting like a lock and key. It first attaches to the cells of the mucosal lining, which is the skin around the nose and the respiratory tract. The virus fuses in the cell and releases its genetic material, an RNA. Normally, a cell would have a factory called a ribosome to produce proteins for the cell to multiply. But now, the virus uses this to produce its own protein, called a replicase, to make more copies of its RNA. Afterwards, viral RNA is cut by an endonuclease, makes its own viral proteins, bought off the surface, and become virions. Millions of new virions are released to infect other host cells, and the host cell dies. And then it spreads to the lungs. By this time, the spikes of the virus on the host cell surfaces are identified by the immune cells, which are like warriors in our body that kill bad cells like viruses and bacteria. Without taking any drugs, someone who has a healthy immune cell could just survive by having their immune cells fight and kill the virus. However, not everyone has healthy immune systems. Once the immune cell sees the viruses, it tags it as a foreigner, kills it, and causes inflammation to let other immune cells know that there has been a bad object they have to fight and kill. However, the inflammation and killing of viruses produces a liquid substance that could leak into the lungs. When a big part of the lungs has been inflamed, pneumonia occurs, making it hard to breathe, so a patient would then need a ventilator. Meanwhile, the immune system continues to fight, and the lungs produce proteins such as a granulocyte macrophage colony stimulating factor, or GM-CSF, to alert more immune cells to come, which causes more inflammations. When the entire lung becomes inflamed, an acute respiratory distress syndrome, ARDS, occurs, and the inflammation and fluid around the lungs prevent oxygen from being transported to the blood, so a person would then be deprived of oxygen. This inflammation and the immune system could also cause a cytokine storm, leading to lung and other organs to fail. So, we really need to develop drugs to kill these viruses. But how are they developed? Traditionally, it takes around 10 years for a drug to be discovered, developed, and approved by the FDA before it can be used. However, we can't wait that long. This is why it is better to find a drug that has gone through that process. Laboratories and companies have libraries of thousands of drugs that they can screen to find ones that exhibit activity against SARS-CoV-2. However, there could be millions of these drugs. This is why artificial intelligence and machine learning have also been used to find these drugs. Ever since, around 20 drugs have been found to have some activity, and some of them have already been approved by the FDA. However, they all still need to undergo clinical trials, so I don't recommend taking these drugs as they could have serious side effects. You want to use them so you won't get killed by the virus, so we wouldn't want you to get killed by taking these drugs without guidance. Now I'm going to give you some examples of the common types of drugs that are currently in clinical trials. The first one is chloroquine. This drug is already approved by the FDA to treat malaria and has been safely used for several years. But how does it work? It is not yet very well understood, but they have found that it could raise the pH level, and it also prevents the development of the cellular receptor, making it harder for the virus to bind and get in the cell. Earlier, we saw how SARS-CoV-2 makes a protein called a replicase to make more RNA. Zinc has been found to block the replicase, so it is unable to make more RNA, but zinc can go in the cell on its own. Chloroquine has also been found to help zinc go in cells. However, chloroquine and its other forms like hydroxychloroquine has some serious side effects including heart problems, changes in heart rate, and vision loss. It is generic and can be bought over the counter in some countries, but please don't take this without guidance from a doctor, as someone has already been reported to die from taking it. Another one is Remdisivir, which was initially developed for Ebola. However, at phase 2 clinical trials, it was found to be ineffective for Ebola, so it was shelved for later use. Recently, it has been shown to exhibit activity against SARS-CoV-2. How does it do this? It binds to the RNA while it is making copies, so it stops making more RNA and the virus stops making more viruses. However, in clinical trials of Ebola, it has been found to increase some liver enzymes that could cause liver damage. So I also don't recommend taking it without guidance from doctors. Both chloroquine and remdesivir are taken orally, and they are distributed throughout the body, so they can also affect other body parts and could cause negative side effects. This is why there's also promise to drugs that only target the lungs. One is Ciclesonide, which was recently tested preclinically by one of the labs I'm working with. It is an inhaled corticosteroid that is used to treat asthma, so it only targets the lungs. As a corticosteroid, it is anti-inflammatory and can prevent a cytokine storm, but has also been found to bind the endonuclease, which cuts the RNA, so when the RNA is not cut, it is unable to make new viruses. Long-term use could cause cataracts and high blood pressure, and it has also been found to interact with anti-HIV drugs, so scientists have to investigate this more in clinical trials. Another drug is Gimsilumab. Since it is a monoclonal antibody, it binds to a specific target. In this case, it is the GM-CSF, which we have seen earlier could cause more inflammation. So Gimsilumab prevents an acute respiratory distress syndrome and cytokine storm. This could be especially helpful for patients with moderate to severe symptoms. This drug was developed in clinical trials for rheumatism, so potential side effects are still unknown, but it is now undergoing clinical trials for COVID-19. As you can see, these drugs all have specific uses, so they could be used for different stages or severity of COVID-19 symptoms. Chloroquine, remdesivir, and Ciclesonide can be used for mild to moderate symptoms, while Gimsilumab might be used for severe symptoms. They all have side effects, some of which could be deadly or harmful, so don't take them without a guidance from a doctor or healthcare professional. I hope you enjoyed this video and learned something new. Let me know if you have comments or questions. Thank you.